Role of selective and differential media in identifying bacteria. Use of selective and differential media in microbiology labs. The learning objectives is understanding our properties and uses of selective differential media, interpret microbial growth based on media reactions, and apply knowledge to identify microbes in mixed cultures, according to Professor Sims. This presentation emphasizes the application of selective and differential media in microbiology. These specialized instruments facilitate the cultivation, isolation, and identification of microorganisms according to distance, growth, and biochemical traits. Our learning objectives are to comprehend the purpose and design of these medium analyzed microbial responses, such as growth and color change, and utilize that information to identify undiscovered organisms. The general purpose is to explain how these media function, while the specific purpose is to show how microbial traits like sugar, fermentation, or salt tolerance can be used to distinguish between species. Upon conclusion of this presentation, you want to comprehend how selective and differential media facilitate precise and effective microbiological identification in laboratory and clinical environments. Filtering microorganisms. Microorganisms require nutrients to grow, such as broth, which is liquid, or agar, which is solid. Environmental and physiological needs like pH, temperature, and oxygen. The classifications is to obligate aerobes, anaerobes, facultative, facultative anaerobes, aerotolerant, and microaerophiles. In order to cultivate microorganisms in the laboratory, the proper conditions and nutrients must be supplied. Culture media, which might be solid agar or liquid broth, are used to supply nutrients. Agar gives all colonies a surface on which to be observed, whereas broth permits widespread growth. Microbes require particular physiological conditions in addition to nutrients. These include ideal oxygen availability, pH, and temperature. The majority of human infections, for instance, are mesophiles, and they thrive at body temperature. Another way to categorize microbes is by how they use oxygen. Obligate anaerobes are injured by oxygen, whereas obligate aerobes require it. Microaerophiles only need trace amounts of oxygen to thrive. Anaerobes can grow with or without oxygen, and anaerotolerant anaerobes tolerate oxygen but do not use it. Professor Sam. Types of culture media. General growth media is the nutrient agar, tryptic soy agar. That supports wide range of organisms. The enrichment media contains growth factors, which is blood vitamins to boost growth or specific microbes in mixed cultures. Professor Sims. Microorganisms are grown using culture media, which differ according to their intended function. Numerous types of bacteria, fungus, and yeast are supported by general growth media such as tryptic soy agar and nutrients agar. These media only offer the fundamental nutrients required for microbial survival and reproduction. They are neither selective or nor differential. Enrichment media, on the other hand, are made to promote the growth of particular organisms, particularly when those organisms are present in small quantities in a mixed culture. They have extra ingredients like vitamins, blood, or other growth agents, as I mentioned before. Enrichment media are particularly helpful in clinical or environmental microbiology because of these ingredients, which aid in the growth of food sensitive microorganisms. Selective media contain agents to inhibit certain microbes, allowing selective growth. The common selective agents contains high salt, pH extremes, and chemicals from Professor Sims. Examples is potato dextrose agar, sabroid dextrose agar, and phenylethyl alcohol agar. The purpose of selective media is to inhibit the growth of some bacteria while permitting the growth of others. They accomplish this by adding selective agents like high salt concentrations, extremely high pH values, um, or certain compounds that prevent undesirable microorganisms from growing. For instance, because the low pH prevents bacterial development, potato dextrose agar and sabroid dextrose agar are both acidic and selective for fungi, including yeast and molds. Gram-positive bacteria are specifically targeted by phenylethyl alcohol agar. It contains phenylethyl alcohol, which stops gram-negative cells from growing by rupturing their outer membrane. In clinical, culinary, and environmental microbiology in particular, these selective media are essential for separating particular microorganisms from mixed culture. Potato dextrose agar, abbreviated PDA, selected for yeast, molds, fungi, acidic, pH inhibits bacteria. Applications clinical fungal cultures, industry monitor dairy contamination. PDA or potato dextrose agar is a selective medium that is frequently used to cultivate fungi, such as molds and yeast. It contains dextrose and potato extract, which provide vital carbohydrates for the metabolisms of fungi. PDA is selective because of its acidic pH, which prevents the majority of bacteria which growing while promoting the growth of fungi. Fungi have a competitive advantage of this medium because they can withstand acidic conditions better than bacteria. PDA is frequently used in environmental monitoring, food industry testing, and clinical diagnostics, especially to find fungal contamination in plant and dairy products. PDA allows microbiologists to isolate and analyze fungal species without being hampered by faster growing bacteria colonies by selective encouraging fungal development. Sabroid dextrose agar, abbreviated SDA, is selective for fungi, which is yeast and molds, and low pH inhibits bacteria growth. The clinical use is the ringworm and the fungal pathogens. The selective medium known as sabroid dextrose agar or SDA is also made especially for the growth of fungi, such as molds, yeast. Similar to PDA, it has an acidic pH, usually 5-6, which promotes fungal growth and inhibits bacteria growth. When it comes to cultivating clinical fungal pathogens, such those that cause ringworm and other skin infections, SDA is especially helpful. Pepstones and dextrose are among its constituents, which offer nutrients specifically designed for fungal metabolism. SDA is frequently utilized in pharmaceutical testing, medical labs, and environmental monitoring, which precise fungal separation is required because of its ability to favor fungal species. This medium aids in diagnosing quality control by separating bacterial flora from fungal pollutants or pathogens in mixed samples. Phenylethyl alcohol agar, PEA, selected for gram-positive bacteria, contains phenylethyl alcohol and 5% sheep blood, and it inhabits gram-negative bacteria. Gram-positive bacteria are the main target of the selective media, known as phenylethyl alcohol agar, or PEA. Its selective Its selectivity stems from phenylethyl alcohol, a substance that damages gram-negative bacteria's outer membrane and prevents DNA replication and growth. When working with mixed cultures, PEA is particularly useful because it suppresses competing gram-negative bacteria while promoting the growth of gram-positive organisms like Staphylococcus and Staphylococcus species. In order to improve the recovery of picky gram-positive organisms and enable further investigations, including hemolysis patterns, this media frequently contains 5% sheep blood. In clinical microbiology, PEA is frequently used to separate gram-positive infections from samples, including wounds or throat swabs that might contain gram-negative flora. Differential media contain indicators that change color based on metabolic activity. Example, lactose fermentation is a pH drop and then a color change. Used to identify coliforms lactose fermenting enterics. Pathogens like salmonella or shingilla. The purpose of differential media is to differentiate bacteria according to their metabolic activity. Indicators including pH sensitive dyes are present in these medium and change color in response to particular metabolic reactions. Lactose fermentation is a typical example. Lactose fermentation by bacteria results in acidic byproducts that lower pH and affect the medium's color. In both clinical and environmental contexts, in differentiating media is crucial for organism identification. For an example, they aid in differentiating lactose fermenting gram-negative enterics called coliforms from non-fermenters like salmonella and shingilla, which are potentially harmful. Biomicrobiologists can determine that metabolic characteristics that potentially identify of unknown bacteria by analyzing changes in surrounding medium and colony color. Eosin methylene blue. Selective function contains eosin Y and methylene blue dyes. Inhibit gram-positive bacteria. Supports growth of gram-negative enterics. In the differential function detects lactose fermentation. Lactose fermenters produce acid, causes dye intake, and then colonies appear like dark purple, metallic green. Non-fermenters remain colorless or pale. Gram-negative enteric bacteria can be isolated and identified using eosin methylene blue agar, a selective and differential medium. Eosin Y and methylene blue dyes, which inhibit gram-positive bacteria while permitting the growth of gram-negative organisms such as Enterobacter or E. coli, given its selective action. EMB differs from lactose fermentation as well. Lactose fermentation by bacteria results in the production of acid, which precipitates the pigments and colors of colonies. Colonies from strong fermenters such as E. coli have a characteristic metallic green sleeve, whereas those from moderate fermenters are dark purple. It's easier to identify non-lactose fermenters since they say colorless or pale pink. Clinical diagnostics and water testing both make extensive use of EM. Mannose salt, agar, selected for gram-positive cocci, Staphylococcus micrococci. High salt inhibits others, Differential fermental fermentation, fermenters yellow color change. The selective and differential medium known as maltose salt is frequently used to separate gram-positive coffee, particularly those belonging to the Staphylococcus and Micrococcus species. It is selective because the majority of their microorganisms are inhibited by its high salt concentration, 7.5%. Additionally, MSA differs in methanol fermentation. It has phenol red, a pH indicator that glows yellow when methanol fermentation produces acid. Staphylococcus aureus and other pathogenic organisms ferment methanol, turning them medium yellow. On the other hand, non-fermenting organisms like Staphylococcus impeditis do not alter the color of the medium. In clinical diagnostics, MSA is frequently used to differentiate pathogenic staphylococci from their non-pathogenic cousins. As far as the references, I got my all of my resources and documents from both MJ and Pierce. It's Microbiology Laboratory Theory, the publishing fourth edition, the textbook, and the open text introduction, and also the notes and the YouTube channel from Professor Sims. Thank you so much, and I enjoyed this season on this class. Thank you.